Chapter 4 discusses the cell, which is the basic unit of life. There are three basic tenets about the cell that makes it the fundamental unit of life. So all living organisms are made of at least one cell. All cells descend from a common ancestor. And all cells share common features, even though they are a very diverse group of organisms. Microscopy is the ability to look at things that the unaided eye cannot see. These are how many things the unaided eye can see. So a chicken egg, a neuron, a human being. Now when we get down to less than close to a millimeter, then we need light microscopy to help us view those items. These are smaller eggs, plant cells, animal cells, prokaryotic organisms. Some cellular organelles we can see with light microscopy. And then when we get down to viruses, ribosomes, proteins, these type things we need to look at with an electron microscopy. So light microscopes use an array of lenses to make small objects visible. Electron microscopes, both scanning and transmission, used to see objects like organelles, those things that are too small to be visible by light microscopes. There are three important parameters when we are doing microscopy. Magnification, so that is the ratio of an object's image size to its real size. Resolution, so the measure of the clarity of the image or the minimum distance between the two distinguishable points. And contrast, so a difference in brightness between light and dark areas of the image. These are all critical in the quality of microscopy that we do. So we have two major categories of cells. We have our prokaryotic cells and our eukaryotic cells. Prokaryotic cells are very small, simple cells. They have no membrane-bound organelles. They first appeared about 3.5 billion years ago, and they are unicellular. They are much smaller than our typical eukaryotic. This is not a comparison in this picture. Eukaryotic cells are larger, complex cells. They contain a membrane-bound enclosed organelles. First appeared about 2.1 billion years ago. And eukaryotic organisms can be unicellular or multicellular. And our cell size does matter because the cell membrane monitors what goes in and out of the cell, helps to regulate homeostasis in the cell. So the surface area to volume ratio is important in the ability of a cell to function efficiently. And so you can see with our total surface area, if we have a 1 by 1 by 1 cubic cell, then our total volume is 1. Surface to volume ratio is 1 to 6. So it's 6. Now if we increase that to 5, now our total surface area is 150. Total volume is 125. And then our surface to volume ratio is 1 to 2. So it drops almost 6-fold, at least 5-fold. But now if we take 5 by 5, so if we take a whole bunch of 1 by 1 cubes to make up the same 5 by 5 cube, now we increase our total surface area to 750. Same volume though, we still have 125 in the volume. This brings us back to our surface to volume ratio of 6. And so we need to keep a small enough surface to volume ratio to effectively transport materials in and out of our cells. So it does matter how large or how small the cell is with how efficiently it can carry out its job. So again, our cells are grouped into two major categories. Our prokaryotic cells, which contain our bacteria and our archaea. So these are the two domains that are made up of our prokaryotic cells. And then our eukaryotic cells consist of protists, plants, fungi, and animals, as we discussed back in chapter 1. Some key features of our prokaryotic cell that are unique are in blue. So a capsule, which is a sticky outer coating, is unique to our prokaryotic cells. Pili, which are these short projections, look like hair all over the prokaryotic cell. Plasmids, so this is small pieces of circular DNA that are found in the eukaryotic cell. And they have a nucleoid region. So this is where they have a coiled mass of DNA. There is no membrane around the nucleoid. It is just a region where the DNA is housed in prokaryotics. Similar features that you will find between prokaryotic cells and eukaryotic cells. Ribosomes, which are our protein factories. So this is where protein synthesis takes place. Our flagellum, which aids in movement. You also have the plasma membrane, and as we will see in upcoming slides, it is made up of phospholipids. So remember our phospholipids that we talked about back in chapter 3 with one of our types of lipids. Cell wall is a rigid outer layer that underlies the capsule of the prokaryotic cell. Also similar with our eukaryotic is that it is filled with cytoplasm. So with eukaryotic cells, we have membrane-bound organelles. We have starting with the center, our command center is our nucleus. This is where all of our chromatin is contained. DNA is the heritable material of organisms. In the center, or in the nucleus, it may not always be in the center, the nucleolus. This is also a structure where ribosomes are assembled. So the nucleolus, or where our ribosomes are assembled. Surrounding the nucleus is a double membrane enclosing the nucleus. So it's a membrane-bound organelle. And it is perforated with nuclear pores. And you'll see when we get to protein synthesis why that is important. It's so that when the genes are transcribed, the RNA can leave the nucleus to come out and join with the ribosomes, be translated into proteins. Directly surrounding the nucleus to capture all the RNA that is coming out of the nucleus is our rough endoplasmic reticulum. And our rough endoplasmic reticulum has all these little dots on it. The little dots are our ribosomes, and that is where protein synthesis takes place. We also have smooth endoplasmic reticulum. It does not have any of the little red dots on it, and it is a little further out from the nucleus than the rough endoplasmic reticulum. This is where you have some carbohydrate lipid synthesis. Also, just outside the rough endoplasmic reticulum, you find the Golgi apparatus. This is where the proteins that are made and the other macromolecules made in the smoothie can be modified, processed, and sorted into vesicles so that they can be secreted out into either organelles, become part of the membrane, or excrete things like hormones and enzymes into the body. Some of the vesicles that are made, organelles, from this are our lysosomes. They contain digestive enzymes. They can hydrolyze things that are taken into the cell or dead or dying organelles. Another vesicle type organelle that we have are our peroxisomes. Peroxisomes are organelles with specialized metabolic functions. They produce hydrogen peroxide as a byproduct and then convert it into water. So these are able to take care of all the free radicals that are made in our metabolic processes and help to keep the body from building up toxic substances. We also have our centrosomes. These are microtubules that are arranged into an organelle. And this organelle we will see when we get to DNA cell division will be important in separating the DNA during making two new cells. And again, as I mentioned earlier, ribosomes are where proteins are made. They can be free in the cytosol or they can be found bound to the rough endoplasmic reticulum. We also have our cytoskeleton. This helps to reinforce the cell shape, allows it to move, allows the membrane to pinch. And so the cytoskeleton made up of our microfilaments, intermediate filaments, and our microtubules. So we have three components, three cytoskeletal elements in our cells. We also can have projections off of the cell that increase surface area, and those are our microvilli. In addition to the flagellum, but not pictured here, is that we also have cilia. Cilia are short, numerous projections that can aid in movement of substances in our body. We find those in our respiratory system and in our reproductive systems. So here are the unique structures for animal cells that you won't find in our plant cells, which are our other main type of eukaryotic cell. So unique structures for animal cells are the lysosomes, the centrosomes, the flagella, and the microvilli. So these are unique for animal cells.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
So that was a nice visualization of our cell, and especially the endomembrane system, which we will talk about again in some upcoming slides. So our plant cells, very similar to our animal cells. The unique features in a plant cell are the cell wall, so this outer structure made up of cellulose, so it's very strong, resist mechanical damage, will keep the cell from bursting. So it is an additional layer to the plasma membrane. So plant cells still have a plasma membrane, but in addition to the plasma membrane, they now have a cell wall in addition to that. Another unique feature are chloroplasts, and chloroplasts are able to take light energy and convert it into glucose. So this is where photosynthesis takes place. But they also still have mitochondria. That is a big thing that happens with a lot of students, is they say that plant cells don't have mitochondria, but they do. They have both chloroplast and mitochondria. And another unique feature of plant cells is this large central vacuole that they have, and they use this for storage. They store waste products, they store water. The enlargement of the vacuole is how plants grow. It is also how they stay erect. So it gives them their turgor pressure. So when a plant's central vacuole is dehydrated or lacks water, that is when you get a wilting plant. So unique structures for plant cells are the cell wall, the chloroplast, and the central vacuole. But then they do have a lot of similar features with plant cells. The rough endoplasmic reticulum, smooth, Golgi, mitochondria. They have the peroxisomes. They have the cytoskeletal elements. So a lot of the same materials as the animal cell.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Okay, so we will stop this video here. We have looked at what constitutes a cell, difference between prokaryotic and eukaryotic cells, and differences between plant and animal cells. Continue to the next video and we'll discuss more in depth the organelles that are found within our eukaryotic cells.